Kenneth Nealson is Professor Emeritus of Earth Sciences and Biological Sciences at USC. Professor Emeritus also at Earth Sciences at Scripps Institute of Oceanography, University of Wisconsin, and the California Institute of Technology. Professor Nealson earned his bachelor's in biochemistry, right? Master's in botany, and PhD in microbiology from the University of Chicago. Okay, where his LinkedIn also points out he was also a very active athlete. All right, football, baseball, swim team. You can talk annual sports with Ken. He also says that he was quote, "I was a kid from Iowa, an Iowa farm who grew up in the '60s in Chicago, and that school, the University of Chicago, taught me how to think." And that's all you want to get out of school, really. Like as simple as that sounds, I think that's really important and cool, but that's on an official LinkedIn, you know, it's very refreshing. Yeah. All right. After his PhD, Dr. Nealson moved on to do postdoctoral work at Harvard University. While there, he received an NIH grant that allowed him to work with Dr. Woody Hastings on something called bacterial bioluminescence. So I guess a lot of this work happened in the Massachusetts campus of Harvard, right? So it was Woods Hole, where Ken and Woody Hastings worked on bacterial bioluminescence. And I guess that was one of the first history-making adventures that Ken went on. After his postdoc, Ken went on to accept a professorship at the University of California, San Diego, at the Scripps Institute of Oceanography, where a lot more oceanographic work went on, I suppose. At some point, Ken wanted to go back to the Midwest because in 1985, he accepted another professorship, a distinguished professorship at the University of Wisconsin Center for Great Lakes Studies. There, he studied how microbes actually breathe rocks. And I'm sure we'll hear a lot about that today, too. Which, of course, that work in Wisconsin laid the groundwork for a completely new field that we know today as electron microbiology. All right, so at some point there, we were lucky enough here in California to recruit him to come work at Caltech as a professor and at the Jet Propulsion Lab up the street here in La Cañada Flynn Ridge as a senior research scientist. And there, he led and established the astrobiology group for JPL. So this group was tasked with a simple task of developing life detection methods for life detection in extreme environments on Earth and places other than the Earth. In 2001, Ken joined the faculty at USC as the Wrigley Professor of Geobiology. And that's actually when I met Ken, soon thereafter that, maybe the late 2000s, where his lab looked at the microbiology of extreme environments and fuel cells. Anyhow, I can go on and on and on. I think our official start time is 30 seconds away. Ken is a fellow of the American Society for Microbiology, American Geophysical Union, the American Association for the Advancement of Science, a member of the National Academy of Sciences Subcommittee for the Solar System Exploration. If that wasn't enough, Ken has a really cool microbe named after him called Bacillus nelsoni that was isolated from spacecraft hardware. It's like one of the most radiation-resistant microbes in the world. And if that wasn't enough for you, Ken's an amazing guitarist and banjo player. I've been lucky enough to jam with him at Catalina Island a couple of times. I'll leave you with this. Ken's work and Ken, the person, have been obviously profoundly influential in my professional and personal life. And I'm glad that he accepted our invitation to come talk to us today. And I hope that he shares his inspirational vitality with all of us today. So with that, Professor Ken Nealson.
Well, he forgot something here. One of my claims to fame is that I'm a member of the Universal Church. And when he got married, I presided over the marriage.
I was the official of my wedding too, yes.
And when Craig Venter got married, I presided over his marriage.
That's right.
So, other than that, I'm pretty much just an old scientist. And I do mean old. Well, I'm going to do a little ridge running today and talk about a lot of different things. I've asked that my talk be recorded and that any of you have access to it. So if you want to go back and look at other things, I talk too fast or whatever, go ahead. This is nothing that's private about this. Okay. I'm delighted to be here. have known about this school ever since I came to Los Angeles, but this is the first time I've ever come to give a talk here, so I'm delighted. You've already heard most of what's on the first couple of slides. Okay, I admit it, I was raised on a farm in Iowa. Okay, and those are how I spent my decade of the 70s or the 60s, I mean. Went there in 61, left in 69. Next one. Did a postdoc at Harvard for 3 years. The bioluminescence industry came because I got to spend every summer at Cape Cod at the marine biology lab with Woody Hastings. Next one. My jobs, yeah, I went to Scripps as my first job, studying symbiosis and light-emitting bacteria. And I still love those things. But I met a bunch of really good geologists at Scripps. My lab was between 2 geology labs. And by the time I left Scripps, I was studying the cycles of metal in the ocean. Because one of these professors had the gall to come up to me after one of my beautiful bioluminescence lectures and say, "That was really a good lecture. Why don't you do something important?" And he's ever a friend of mine because I asked him what it was important, and he said, "We know nothing about iron and manganese in the ocean, except everything goes too fast." And I said, "Well, it's a bacterial then. That's what bacteria do." So enough of that. When I went to Wisconsin, I went to the Center for Great Lakes Studies and virtually studied nothing except iron and manganese metabolism in Midwestern lakes. And that turned out to be really important because there's almost no sulfate in freshwater. But the ocean is full of sulfate. And it makes all this sulfide when you take away the oxygen. And it dissolves all the manganese. So I never would have found this stuff if I'd have stayed in the ocean. Okay, it's almost, so next one. Then I got an offer I couldn't refuse from Caltech and JPL to come work on one of the Mars missions. And this has a little hilarious story to it that I gave a lecture and one of the NASA people was there and I showed a picture of Mars. And I said, "I don't see why they say there's no life could be there because there's no oxygen. My bugs would just use the iron oxide that's on these, and they use it for respiration instead of molecular oxygen. And they use both iron and manganese oxides. Extremely, almost everywhere in freshwater, as long as there's an anaerobic site. Okay, if there's any oxygen, they'll prefer that. But if take away the oxygen, they'll start using other things. And luckily, we discovered these bacteria that use manganese oxides as a terminal electron acceptor. Okay, next one. And then this wonderful job I was offered disappeared when they canceled my mission. And so luckily I got a job at USC, and that's where I was until I retired a few years ago. Next one. And today we're going to talk about non-Earth-centric life detection. And why is that interesting? Because NASA needs to start thinking about how you would find life if it wasn't like earthly life. Virtually all of our life detection tricks have to do with specific molecules that all of life has on Earth. Okay, DNA, RNA, specific proteins, certain membranes. We know all those tricks, but what if there was something hiding out there that didn't have those in it? And that was when I was at JPL, that's what we were working on. And then I came to USC and we had to change directions a little bit. So next one. Some of you probably wonder what is geobiology. I'm accused of starting that field and I had nothing to do with the start of it. Okay. I just picked up by reading the old books and seeing the questions people asked when they were writing their papers. Okay, so next one. In my definition of geobiology, it studies the way that life alters the earth. Okay. The atmosphere, the hydrosphere, and the lithosphere, you all know what they are from top to bottom. And all the water. They're not complete without the biosphere. Okay. And next one. And this is the study of how it has occurred on the planet we know that has life. We can't study geobiology anywhere else yet because we haven't found life anywhere. So almost everything that NASA does is to look at the geology of the system. Okay, next one. And this is probably the best example of coevolution that I can think of. Okay, if you study symbiosis, you know that it's the host and the symbiont co-evolving through time. But here's a whole planet and the life on the planet co-evolving. And it would look drastically different if there wasn't life. Okay. And if any of you have ever read any of the books by, what's his name? Called the Gaia Hypothesis. How many of you have ever heard of that book? Gaia, which in Greek means the Mother Earth. Okay. And he talks about the evolution of the planet. And you do a modeling experiment with Mars. And you say, here's how big it is, here's the gravity, here's how much atmosphere it should have, here's the temperature it should be from the sun. And it comes out exactly like it should be. You do that for Earth, and every gas in our atmosphere is off by at least a factor of 100. Okay, this is a magnificent view of what coevolution is. And these are problems, not problems, they're facts of what life has done to our planet. Okay, and it's coevolution. The planet does stuff back to life, that's for sure, and supplies lots of stuff. So next one. And I want to look at, just to refresh some of your memories and maybe give some of you a new memory. I want to show the next slide. And look at coevolution on the planet, of our own planet, as it's viewed by some people. Next one. And it really is an issue of time. So much of us talk about time. Most of us don't really have a good sense about time. So the next one. So here's a nice diagram of the oxygen concentration on the planet over 4.5 billion years. Okay, we've been here a long time, we being Gaia. And what happens if you look at the oxygen concentration is somewhere between about around 2 billion years ago, between 2.5 and 2, all of a sudden, the oxygen level shot up 1,000-fold. This didn't happen because the planet was evolving. It happened because cyanobacteria appeared. Okay. You can imagine all of this life down here was running like hell to get away from the oxygen. Really toxic molecule because they had no way of processing it. So this is when all hell broke loose on our planet. And it looks like a straight line, of course, but this is 100 million years. Okay, so that's what you have to get used to looking at a graph like this. Next one. And those years, from about 4 billion over to about 2 billion, were the years of the prokaryotes, the bacteria, the archaea, the cyanobacteria. There was nobody else here. Okay, next one. The first eukaryotes didn't appear till around 2 billion years ago. Okay, so now the coevolution is occurring. You have an electron acceptor, oxygen, that you get so much energy out of when you respire it, that you can start developing really complex organisms. And this is where they start to appear. And the complexity is in their metabolism, not so much in their shapes. Next one. Multicellular life didn't really appear until about 500 million years ago. And we think of these things, you think of the one to the Natural History Museum. And they say, age of life. And it's over here someplace because they go back to about the dinosaurs, okay? Here's the ancient life. If you want to figure out how the earth really evolved, this is where you need to look. Next one. First invertebrates didn't appear till here. Next one. Dinosaurs over here only a couple 100 million years ago. They're newcomers. They're the babies on the planet. Next one. And here's man. They say if you put your arms like this, and that's the 4.5 million years ago, and you take a nail file and you scrape off the edge, that's how long man has been here. Okay, so we're awfully proud of ourselves for that. But overly proud, I'm afraid. But anyway, I wanted to start here because we often forget this. Okay, and if we're going to another planet, it would be really nice to know where we are on this graph. Because if you landed on Earth back here, there would be plenty of life, but without a microscope, you wouldn't have known it. Okay, and these are really critical things that you need to think about over time. And it's so easy to not think about them. So next one. A few thoughts that I had that I wanted to share with you. First, that geobiology considers the geosphere over time and space. Next one. And life is intimately connected to this geosphere. Okay, there are so many connections, especially in the microbial world, where the geosphere is supplying both the electron donor, the food source, and the electron acceptor. Those are geological products, except of course for oxygen. But remember, except for eukaryotes, there is no other group that absolutely requires oxygen. Okay. Now there are bacteria that are aerobic, but there's a whole part of life that's intimately connected with this. Next one. Energetically, it's connected. Physically, it's connected. Bacteria live on rocks, they live in rocks, they live under rocks. Okay. The minerals on this planet are connected to where a lot of life lives. Next one. And chemically, of course. Next one. The other one is to get back to the time issue. Geology goes slowly. If it went really fast, it wouldn't be a good place for life because all of the energy would disappear before you could harvest it. Okay. But these are factors of 1,000 in terms of speeds, sometimes more than that. And that allows life to harvest a lot of the geological energy that's here. So, and biology is famous for speeding things up. Okay. Our bacteria make catalysts that take a chunk of manganese oxide, which should have been stable for 10,000 years, and completely dissolve it in about 18 hours. Okay. And these are the tricks that life has learned to do to get energy out of the environment. Next one, enzymes, catalysts. And that the whole business of time isn't something we should forget. Okay, next one. So I want to now go some of you will want to shoot me after I do this, but it's important that we talk about this, that there are lots of fuels of life, right? We know about food, it's glucose and all of those carbohydrates that make us look like we do. And we know about oxidants, but here's the fuels. In organic carbon, there's a huge variety. And what I've done here is plot the relative voltage that you would get out of the same amount of any food in organic carbon. But there are bacteria that live on hydrogen sulfide, elemental sulfur, iron, ammonia, and even manganese. And they do this by stealing electrons out of the hydrogen in this one, the same way our enzymes take the electrons out of our Coca-Cola or sugar or whatever we're eating. And they pass those electrons over to the oxidants. And that gives you the energy to run life. Virtually all life on our planet runs this way. Okay, and this is something I almost forget to teach you in biology class. Okay, so, and next one on the oxidants there. There are oxidants that can be organic also. The best oxidant is oxygen. Okay, if you take an electron here and run it down to here, you get this much energy out of it. So if you're growing on oxygen, you have a good deal. Okay, you can get a heck of a lot of energy. If you take away the oxygen, we all go, okay? In fact, every eukaryote that we know, except a few strains of yeast, will die when you take away the oxygen. That's why we all look out for cyanide. Okay, it stops the oxygen uptake. But if you do this in its bacteria and the sediments, some will use nitrate, some will use manganese oxide. These are my bacteria. Selenium oxide, iron oxides, arsenic oxides, sulfate, and even CO2. There are bacteria that can grow on any one of these things and respire and make enough energy to make another copy of themselves. When we go to a new environment, we measure these things and we measure these things, and then we know what not to do, what not to bother to look for. Wouldn't that be nice if that's the first thing you did on another planet? Because this is almost a universal way that you get energy out of the geosphere. You steal electrons and you use those for energy. Now, next one. As I said, this is the voltage available for charging up the biological system. Next one. If you take any one of these fuels and hook it up, in this case to CO2, there's only a couple groups of bacteria we know of that'll do this. And mainly they're methanogens. They're bugs that grow really slowly, getting a tiny bit of energy, and they make methane. Next one. And next. The iron reducers get a little more energy. They grow faster, they do well. Next one. The nitrate reducers and the manganese reducers are about the same. And finally, oxygen. The message for this is over here. Oh, the geological environment, as you can imagine, supplies almost all of these. Okay, when the stuff comes out of the hydrothermal vents in the middle of the ocean, it's mostly in the electron donors. By the time you go to the ocean for a swim, it's mostly the oxidized parts. Okay. One of the reasons I like to show this to everybody is you can take it home and stare at it. But if you, looking at the energetics here, you connect any of these fuels with any of these electron acceptors, And there will be a group of bacteria that grows by that mechanism. Now, isn't that remarkable? You know, that they have figured out every energetic niche on our planet. And you would expect the same would be true of any planet. What they really did, though, was figure out how to make oxygen. And then if oxygen's there, it can chemically oxidize some of these things after they've been reduced. So oxygen is the big motor that runs this system. If there's a downward slope on that arrow, there will be a bacteria that lives that way. And life has truly evolved on this planet to take advantage of that. The major place you see it is in a sediment. Okay. Carbon dead things come and land on the sediment. The bacteria use up all the oxygen. And when the oxygen's gone, you'll see layers in the sediment as each one of these things is used. And the bacteria are smart. They use them in the right order. All the oxygen will disappear, then the nitrate will disappear. All of these things in order. They're metabolically very shrewd. Okay, they don't need brains to know this. They just need a few energetic symbols. Next one. So that was a heck of an introduction, but in some ways, if anybody has questions or wants to talk about any of this stuff, I'll be happy to talk about it. If I don't get by the fourth slide, it's okay with me. So I promised we would talk about Mars. Why would you want to go study Mars? And a couple of small points. A huge amount has been written and movies made and talked about Mars. And so, and it all really started back in the 1800s with Schiaparelli, who was an Italian astronomer. And he got a nice big new telescope. And he reported that he could see continents and seas on Mars. And wrote several books about this. And people started believing that there had to be life on Mars. And then along came Percival Lowell from the Massachusetts area. I think he was a professor at Harvard, but he was also a personally very wealthy man. So by the time he had retired, he had built his own telescope facility in his backyard, I suppose. And there are great pictures of him. And he wrote a book called Mars as an Abode for Life. Okay, and it had pictures of the rivers and the canals and all of this kind of stuff. And unfortunately for him, telescopes got better. And as telescopes got better, I should say first, some of you, if you don't know this, look it up and read about it. That Orson Welles in 1938, largely influenced by Percival Lowell, had a fake radio announcement that Martian aliens were invading the US, that they had landed in New Jersey, and they causing all kinds of trouble. And it scared a major part of the population. And it became really famous because he had to figure out how to stop it. You know, people were driving away from New Jersey and doing all kinds of stuff. And in those days, you didn't have very fast cars. And everybody had a radio. You know, almost everybody had it on at night. And it evidently just caused a huge holocaust of fear and running. I hasten to think how many lawsuits would be filed today if that was right. You know, it just would keep the courts busy for 100 years. Anyway, this was part of the scene. And I mention it because it was on everybody's minds. You know, and Mars, it's easy to justify studying something like this. Next one. And in the 1970s, Viking mission went to Mars. And they not only landed successfully, but they did some biology experiments. And I'm not going to go into the details of those. But one of them became really contentious. And the lead scientist was someone named Gil Levine, who claimed that he found life. All the other scientists looked at the controls on these experiments and said, no, the Mars soil caused this effect. And it really came by adding more of the inoculum the next day. And if this was life, it would have gone even faster. But it didn't go at all. And so there was, but poor old Gil, even as late as 2016, he had a paper in the astrobiology journal, again saying, why don't you people listen to me? I discovered life. Okay. It turns out that the consensus for this from all the other scientists was that the soil on Mars is very, very oxidizing soil. And when you add some carbohydrates to that, it just oxidizes them and releases CO2. And of course, it was radioactive, and the counters saw the CO2 and the radioactivity. I was actually listening to that on the news in 1977, I think. And I got so excited. And then the next day, all the excitement was gone. Okay. But those are the kind of things. And when I should mention that Gil Levine was also independently wealthy. And so you couldn't shut him up. You know, he had enough money to buy his own journal and publish the papers. And so you have to be a little bit tricky about something like this. Anyway, this is where I joined in. And there are Martian meteorites that have really gotten us into trouble. We can tell when a meteorite comes from Mars. If anybody wants to know, I'll tell you now. Okay, but when the meteorite is initiated, it becomes from the planet getting hit by a meteor and all of the stuff turning into liquid and being launched into space. So a big meteorite hits Mars, maybe a couple 100 tons of soil and stuff gets launched into the Mars system. And it has holes in it, and it picks up atmosphere of the Mars, and then it hardens. So whenever a meteorite lands on our planet, they take it to the right analysis system, they drill a hole and sample the atmosphere. And it turns out we know exactly what the atmosphere of Mars is from the Viking missions. And you can say, yes, that's a Martian meteorite. And every other mission that's gone to Mars has affirmed this. Okay, we know the Martian atmosphere really well. And there were some scientists, good friends of mine, who looked at one of these meteorites and claimed that they saw the remnants of bacteria. And this was going to prove that there was life on Mars. And it just wasn't so. Okay, when you really look at the data they showed, it was not at all convincing. And it was much more convincing that these were contaminants from earthly life. Because these meteorites fall and maybe stay on the Earth for 100, 1,000, 2,000 years before somebody picks it up. And even when you pick it up, unless you have sterile gloves, everything is a source of contamination. And between the contaminants and some of these things that they didn't have, what do you call them, bars on to tell, and they were about the size of a ribosome, not the size of a bacteria. And so, but they did look interesting. Okay, but anyway, these guys are no longer friends with me. Okay. And yeah, that's the way it goes sometimes. Okay. Practical reasons. This is one of the only places that's close enough to go to. Okay, we can get to Mars in only a year. How would you like to go to Jupiter and take 8 years? Or maybe go to Titan and take another 15 years, probably. The solar system's a big place, okay? And one of the Mars missions just got canceled because it was too expensive. Okay, okay, that's what happened to me when I was at JPL. My mission got canceled. Now, 20 years later, it's happening to another group. But even Mars is too expensive. It's too expensive. It's a great place to go. Over the 30 years or so, well, from the 1970s onward, we've had successful missions to Mars. Okay, that's 50 years of experience. As we were talking about at lunch, most of the early missions to Mars were failures. Okay, they got close. Every once in a while, they got close to Mars, and even a couple of the Mariner missions flew by to take a close-up picture of only 1,000 miles away or something. So over the time, now we have mobile laboratories crawling around the surface of Mars. We're doing fantastic things, but every mission builds on the mission before. And it may 2 or 3 years, you think of 2 or 3 years for each mission to learn one little new thing and to hope it lands successfully on Mars. This is just amazing technology. Next one. And we know a lot about the planet. So far, there's not a single suggestion other than Gil Levine that there's life on Mars. Okay. Now that's almost as good as discovering life, because you want to know the truth. Okay. And if there isn't life, Mars becomes a really interesting place to study, to look at geological evolution. Okay, because planets do evolve. The hot core gets colder, they change, they lose their atmosphere, things happen. But it would be way nicer to find some life on Mars. Okay, next one. So, life detection. Is there or is there not, or was there not ever life on Mars? And you can ask what needs to be done to even ask this question. And often we forget to do that. Get to it. Next one. We have to define what life is. How can you find something if you didn't define it and say, this is what we're looking for? Okay. And nobody ever told me that when I went to JPL. They just said, you got to go find life. So we put together a group that we called the non-Earth-centric life detection group. And the question is, next one, you have to decide what to measure, okay? And if you can't decide what to measure, you certainly don't want to do just the usual earthly things. We probably got 20 proposals a year to send a DNA machine to Mars. And it only cost a few $1 million to send that experiment to Mars. But it isn't the way you should look for life. You make that assumption, you're almost for sure not going to find it. So DNA, RNA, specific proteins, lipid membranes, you go on all the way us microbiologists use to detect life. They have a big X through them. Okay, so next one. And you'll probably need to develop some new methods because you're out talking about measuring new things. Next one. And we may actually have to bring samples back. Because if you want to do some giant machines in a synchrotron, which has 5 miles across or so, you probably won't fly that to Mars. Okay. And so many of the things that have the level of resolution we want are big machines. They'll fill up a laboratory with a single machine. So maybe what we should do is focus on bringing samples back. And that's what we're trying to do. Next one. But if you bring samples back, now you have a planetary protection issue. What about all those people that saw the Andromeda strain? And all of these horror movies about things that are going to destroy our civilization. When I was at JPL and I was the chief scientist with the sample return mission, my major problem were all the people who wanted to make sure we didn't bring back the samples. And they even proposed sending them to the moon and setting up a laboratory there to sample. But they were terrified of these things. There's a whole society against Mars sample return, which is a very quiet society until you say you're going to bring back a sample. Then the phone just rings every half hour to tell you, you're crazy. I know there's life there. There's some, yeah. Did you have a
It
question?
kind of sounds like Venom from Spider-Man or something. It
Oh.
sounds like Venom from Spider-Man. Whether if you're like someone's gonna bring back Venom or the symbiote.
Say again.
Oh, you think like someone's gonna bring back Venom or the symbiote from Spider-Man?
Well, they don't know what they're looking for. They're just afraid of it. I mean, we don't, if you saw the Andromeda strain, if you haven't seen it, look it up somewhere and get an old movie copy. It's worth seeing. It's a really scary movie. And in that case, it was an organism that could kill humans really fast. And a venom probably wouldn't be any danger at all because it would kill a few people that it happened to get into. But unless it can grow and keep going, live off of us humans as an energy source. But heck, lots of animals do that. That's what Darwin's theory is all about, survival of the fittest, right? So this whole business of planetary protection is a two-way street. You need to protect the Mars and the samples from Earth contamination. You need to protect the Earth from Mars contamination. And you have to do both things if you bring a sample back. Okay. And if you talk to an engineer that builds things like chambers, it drives them crazy because you have to have positive effect to protect the sample from the Earth. And you have to have negative effect to protect the Earth from the sample. Now you tell me how to do that, okay? But the engineers at JPL had it figured out. Okay, next one. Just more of the same stuff. And planetary protection is a big deal at JPL. Next one. So this is what we said before. How would you measure or find life if it wasn't like ours? Next one. So then we have to say, we want to meet once every 2 weeks and come up with the universal properties of life, whether it's like ours or not. But we can, now I'm going to give you a clue. Who knows what some of the, this is a test, you'll all be graded. What the universal properties of a living organism would be. That's good. You have to speak out loud. My ears aren't very good.
Hydrogen, oxygen, phosphorus, and nitrogen off the top of my.
No, you're talking about individual chemicals, okay? I'm talking about if you just came to a place and said, is there anything here that's alive?
Some kind of metabolism.
Yeah, yeah. And what about metabolism? What does that imply?
It's changing the properties of the
Okay.
environment. Something.
It's got to eat something, right? And if it eats something, what's the other part of metabolism?
Waste.
Waste material. Okay. You had a suggestion?
And this is maybe too basic, but something that is not in energetic equilibrium with its environment. Like you kind of have to like use energy, right, to stay out of it. If you're at equilibrium, you're covered with that.
Yeah, it's in,
It's the same thing.
yes.
Proliferation.
Proliferation, it should grow. If it doesn't grow, it's probably not life, right? At least you couldn't tell it from no life. Any others? Sorry, again. What did she say?
The ability to reproduce.
Yeah, you can divide growth into both reproduction and getting bigger. So it's part of the business of getting. It should change size, and either the population changes or the size of the organism. Yeah,
I'm not very familiar with biology, but would also cell replication be one of those by chance?
say again.
Cell replication, like cells replicating themselves?
Yeah, so again, it's part of the growth thing. Okay. One more. Yeah.
Kind of like, it would be outside of energy delivery, there should be a boundary between the system that we call a unit of life.
Yeah, well, it should take energy from the environment, right? And the unit. I don't know how to say that in other words, so I'll leave it with your words. All right, so this is the kind of stuff we used to talk about once a week, once every couple of weeks for a whole year. So I don't expect you to get all the answers next one. When you get those things, what is it possible to measure? Okay, now you had some good ideas. You had input of energy. That's measurable. What about the waste? If you know what the waste is, you can measure that. If some other organism doesn't eat it before you measure it. Okay, so keep going here. And is any of this possible to do without bringing samples back? Next one. So keep on going here. This is a little bit of a repeat. Okay. I think I had those out of order somehow. Next one.
Yeah. keep going.
So what are the things that we came up with? Okay, next one. You got this one right. Uses energy from the environment to sustain itself. It's sort of a thermodynamic definition. Next one. Reads waste products. As it metabolizes, this is also a thermodynamic issue. Next one. It alters the rates of things. This is what the marine chemists told me, that's going too fast in the ocean. Okay. To me, that's proof almost that there's some kind of biological catalysis. Next one. It has a complex and unique chemistry. That's what a couple of you were talking about. And where you started out with the phosphorus and the other things. What if I told you that all the life on our planet has the same ratio of nitrogen to phosphorus and what's the other one? Carbon, yeah, carbon, nitrogen, and phosphorus, the CNP ratio. This was discovered only in the 1920s or something, that no matter where you took life from, samples from, and you looked at the CNP ratio, if it was alive, it had that ratio. There's no mineral on this planet that has that same ratio. Isn't that remarkable? No mineral that we know of, all these thousands of minerals, and none of them has the CNP ratio that life has. So we would say, well, isn't that interesting? We should just find something that looks like it. There's a lot of it, because there are a lot of them, and measure the elemental chemistry. And if it doesn't have to have our CNP ratio, it just has to have a ratio of elements that's different from the minerals on the planet. Okay, next one. And it has complex and unique chemistry. Next one. Reproduces. You got that right, okay. It adapts to changes over time. Okay, it evolves. There's evolution of these organisms as well as of the planet. And it should be able to move. Isn't that interesting? Everything we know about on our planet, except maybe viruses when they haven't got inside of another organism yet, shows non-random movement. Sometimes you have to look inside the cell to see it. But you know, a physicist once argued with me that trees don't move non-randomly. And I said, well, how did the water get from there up to the top of the tree? He said, and it's true. I mean, everything that we do has non-random movement because it's the complexity that you have to deal with. Next one. And it almost certainly will alter the geosphere. Okay, because it'll be taking up these elements and using them in ways that wouldn't happen if the life wasn't there. Next one. So I'm going to end with this, even though there's a lot of other good stuff. And we'll have our life. And one thing I left out here was that it should have a shape. What if you had life and there weren't any walls around it? All the energy would just diffuse. No matter what you did, you couldn't have, you have to have a shape. And certainly on our planet, there are no organisms we know about that don't have a shape. Okay. Should be, have a lot of complexity. Next one. Energy should go in. Waste should come out. And these can be biominerals, they can be other cells. And movement will be assessed both here and there. Next one. They should replicate. Next one. They should evolve. Now, next one, which of these are measurable? I don't think replication is measurable. We have places, well, if you have enough time, it might be. But there are places on our planet now in the deep sea where organisms double every 100 years. So if you have a relatively short mission, you don't have a chance of making this measurement. Okay. And what about evolution? Again, it's the same problem. We don't know the time scale for evolution. But if you see a lot of different shapes and a lot of diversity and many copies, you can infer this is going on. You just can't prove it. Okay, next one. The approaches then, structure and chemistry. And there will be a lot of words here that some of you don't know. I've only got 2 minutes left. It's thermodynamics and kinetics. And the next one is movement. Okay. And the last thing, I think on the next slide, it brings up one last thing. You need a solvent. Okay. Chemistry being done with solid materials has to be heated up to hundreds of degrees to make it do something. Organisms do this just by being organisms and having enzymes, but you need a solvent. On this planet, it's water. Could there be another solvent that could be used? Are there places in our solar system that have lots of water? The answer to that is yes. The moons of Jupiter, Europa, Ganymede, and Callisto, all have more water than the Earth has. And they're moons. But they're basically ice balls that have liquid water inside of them. And that's why people get so excited about sending missions to Europa or these other planets, other moons. It's just really hard to land on Europa. And there's a lot of gravity from Jupiter that gets in the way. And it's tough. What about other solvents? Titan, one of the moons of Saturn, has oceans that are ethane and methane, liquid. I think there are 32 giant lakes on Titan that have this, and the boiling point of ethane and methane is about a minus 150 degrees centigrade. And the freezing point is about 90 degrees. Oh, sorry. Got this wrong, but yeah. Minus 90. And so there's about 100 degrees difference between the boiling point and freezing point. And isn't that interesting? That's just about the same distance as water. So couldn't you manage? Now you have to be a real chemist because you have to figure out what the membrane's like in an organism in liquid methane. But that's a great thought problem. And one reason for going back to Titan, as far as I'm concerned, because it's so far, the only other planet with an accessible solvent. To get to the solvents on Europa, you have to go through a couple of kilometers of ice. And, but this would be on the surface, so it has access to sunlight. There's all sorts of interesting things you might talk about Titan. Okay. Anyway, I think I have another 10 slides, but this is exactly as I said. I didn't care if I got through all this, and thank you.
Thank you so much, Ken. Obviously, I think we're scheduled till 4, so we're a couple minutes over. But if no one comes in the room, I think, can we be open to entertaining maybe an additional 5 minutes of questions?
Yeah, I'm here. I don't go home till he leaves because he's taking me, he
It's my hot seat.
agreed to give me a ride. So, yeah.
So I guess I'll open up the floor. If anyone has to leave, that's totally fine. I understand that we're scheduled till 4, and I don't know if anyone else is coming in, but if no one is, let's take 5 to 10 more minutes for questions. If anybody wants to ask questions, feel free to do so now.
And I apologize. I didn't bring my hearing aid today, and that was a stupid mistake. So I may need an interpreter if anybody asks a question. Yeah,
I'll jump in and interpret.
okay.
Yeah, I mean, it's hard to be a question, but everything still is based on carbon, nitrogen, oxygen. Is there a possibility that there are different elements out there, like different chemical structures?
So the question was, is it possible there are different elements? And I think you have to give Mr. Mendeleev a heck of a lot of credit when he put together the periodic table. And the only place there's any room for other elements is in the really heavy elements. Okay, because you can extend the periodic table a couple of lines further. There are more the nucleus just gets bigger and bigger. But when you have them, they're harder to handle. And it's really hard to think about life evolving with the big heavy elements in. So luckily for us, I think we have the first couple rows of the periodic table on each side and a lot of transition elements in between to carry electrons. And it makes sense that life should somehow figure this out, okay, which it clearly has on our planet. The question is, what about all those other billions of places? And how the heck do we find out? Sometimes we don't see the image for 20 years after it's happened, 20 light years away. And we think it's really good getting to Mars, okay, which is only 100 million miles. So I think we will answer some of these questions in the next 50 years when our robots get so good that we can send them on the spacecraft and don't have to worry about feeding them and cosmic ray damage and all the things that happen in spaceflight to humans. Why not have an avatar kind of system where you as a human are living inside that robot who isn't subject to all these things. You could just let them sleep for most of the mission and then wake them up when you get there. some of you will live long enough to see if there was any truth in this. I won't, but, and during my days at NASA, we were talking at lunch, people hated it when I said that. Because the whole astronaut, the aura of the astronauts is something that drives NASA. And they don't want robots to do this, and they were very vocal about that. Yeah,
So since it's really hard to come up with something that it's non-Earth-centric for life detection, what if we try to grow whatever life we have here in Mars and see how that behaves and how that reproduce, like cell division, all that, and then maybe we can have an idea of the thing that we know, but how it grows over there in terms of like gravity.
well, yeah, that's a really interesting point. And that's a point that was pretty seriously considered in the 1950s and the 1960s, I would say. And then a microbiologist working at Yellowstone National Park discovered, bacteria that could grow at 100 degrees centigrade. They could grow in boiling water. So at Yellowstone, doesn't quite get to 100 because of the altitude, but this whole business of extreme life on our own planet has greatly expanded the view of where life could actually be. And if you try to restrict that by saying, well, what can't earthly life do? That border keeps moving every year. Okay, we now have bacteria, high isolated bacteria that grow at -20 degrees. Okay, and they just do that in layers where there's enough salt water that the water isn't frozen yet. So if water is your solvent, you can push these limits of life a long ways. But when it becomes either a solid or a gas, that goes away. Okay, so a lot of the really good hot growing bacteria are in the bottom of the ocean, where you can get water up to 200 degrees centigrade that's still liquid. Okay, and so that's a good way to think about it. But then what that does is make you go to the earth and see, well, are there places that are there? And the answer is yes. They used to have something called the habitable zone, and it was a distance from the sun, and on the other side, the distance from the sun, and over here all the water was frozen, and over here all the water was gas. So that's a good, now if you use that same approach on Mars or Titan, you can again get the limits, and it isn't like earthly life. Okay, because one of them is way out in the solar system, almost no sun, and the other is, or Venus, where everything's 700 degrees or so. Yeah.
I have a question about Titan. So there's this water on Titan that was supposed to be frozen, but compared to Earth, why does it have ethane and methane in the water compared to Earth?
So I'm sorry, my ears
Oh,
are
it's
so
okay.
bad.
Sorry for having loud. Why does Titan's water have ethane and methane unlike Earth? What causes that?
Oh, because it's so doggone cold. Okay, that's the boiling point of ethane and methane. Those were minus Kelvin, they're minus centigrade temperatures. And so there's no liquid water anywhere on the planet because it's so incredibly cold. But ethane and methane, if you get them in the right mixture, they're quite good. And they'll form a nice liquid environment from about -90 to -190 centigrade. And now you have to figure out, you have to be a much better chemist than I am Because on our planet, we have lipid membranes that keep the water out. Okay, and keep the other water in. And if you didn't have a way of controlling this salt, you couldn't have life. Okay, so what the heck are the membranes made out of in an organism that's growing in liquid methane or methane? Fantastic. They might be some. Now you have to go to the industry and say, well, there are some silicon polymers that are really good at this. Okay, that's why we use silicon to make our doors waterproof. Okay, well, you know, start being an out of the box chemist here and say, well, I'm going to make my organism that grows on methane and methane, and it's going to think that's just a perfect temperature. And remember, to get energy out of the environment, you just have to speed things up a little bit. So all they have to do is make mechanisms. And then you go and say, well, what else is on type? And that's a very tough question, because it's good enough that we know that there's methane and methane lakes, but we don't know much else, because we weren't expecting that when we went to type it. So do we want to spend another $1 billion to find out what else is there? We're not afraid of any of those organisms because they would die instantly if they got to earthly temperatures. So, question.
Final question.
Sorry.
Yeah, no, you're good. Yeah. No,
This is kind of, this is a really random question, but you did botany, so it makes sense. So we talked about like evolution, right? Adaptation being one signature of life. I wonder if you could, instead of having to bring things back and like wait to see if they're adapted or whatever, since like getting back to the Gaia hypothesis, right? Like life changes the physical and chemical properties of its surroundings. Could you do like a reciprocal transplant experiment on Mars? Like, are there different environments on Mars is what I'm saying. Could you take a sample of rock and dirt from one spot in Mars and put it in a different environment and do the same thing, swap the other one, and then see if their chemical physical properties change because they changed in Mars environments? Would that suggest that there might be something?
That's an interesting question. One of the things on my later slides was, what do we really know about Mars? And what isn't on that slide is to answer why we know so little. And that is that it's really hard to land on Mars. And so you land on all the safest places. But all the interesting places are those not so safe places. Mars has a Grand Canyon that's 3,000 miles long. Okay, and they don't even know how deep it is. Okay, now there's a place where you can get to the subsurface. You'd be looking at the side of the side walls of a giant canyon. Mars has a volcano that's as big as the state of Alaska. It's just Mars, and that probably means that virtually all the heat from the center of the planet comes out of one place. Okay, where on Earth we have these vents all around. We have 9 plates with vents coming out, 50,000 miles of mountains under the ocean that are from these spreading centers. Okay, completely different from what we have. And as much as I would like to say that's a good experiment, until we get a lot more technology, it's an impossible experiment. And I think it's an experiment if you saw the movie The Martian. If you didn't, you should go see it because it's pretty good. They got most things right. But they totally ignored the existence of the interesting places. Now, the mission that's going on right now with a rover called Perseverance, they have a helicopter on. And they've learned a huge amount from this helicopter going out and taking pictures and coming back. It did more in one day than most of the missions have done in 2 years. Okay, in terms of going someplace new. Couldn't do experiments, but that's a remarkable feat because the atmosphere of Mars is only about 1% of what it is on Earth. So how the heck do you get something to fly without any atmosphere? And we did it, and it's really exciting stuff. What that could do is set the stage for your kind of experiment, because the helicopter would say, okay, I can go 20 miles over there and back. Could it carry one of your rocks over and drop it off? We have drones that are smarter and can do things. Answer has to be yes, but the technology is lagging way behind the ability to ask the questions, I think. And, but it's way better than it was 10 years ago. So
Well, on that note, I once again want to thank our speaker, Ken. Thank you so much. Fantastic experiment. I can't wait to see the day when we can transform a polar rock to the Martian tropics, right, and vice versa, and seeing what happens. Yeah, so thank you all for attending. We will see you again in 2 weeks, I believe. So fantastic. Thank you so much, Ken.
yeah.
That was a treat. Thank you. Thank you.
I wanted to ask your opinion, where do you think most confident?
Let's start working on that proposal.
Apologist, so.
Let's start working on that proposal.
Titan?
When you're
It's just, it has an organic